Characterizing the transport properties of therapeutics and their carriers is critical for ensuring effective biological responses. These methods aid in designing optimally charged drug carriers for targeting negatively charged tissues. The main advantage of these techniques is their ability to better predict in vivo therapeutic efficacies through a series of in vitro experiments that characterize solute transport through tissue.
Cartilage diseases such as osteoarthritis remain untreated due to the inability of drugs to penetrate the dense avascular cartilage matrix, requiring drug carriers to prolong therapeutic efficacy.
Begin by using a delicate task wipe to gently remove excess PBS from the surfaces of 3 mm diameter, 1 mm thick cartilage explants. Use a balance to quickly record the wet weight of each explant and immediately place the explants into a PBS bath to prevent dehydration. Next, add 300 microliters of freshly prepared 30 micromolar fluorescently labeled cationic peptide carrier solution per well to the inner wells of a 96-well plate, and use a spatula to add 1 explant to each well of solution. Fill each of the surrounding wells with 300 microliters of PBS, and cover the plate with a lid. Seal the edges of the plate with flexible film to minimize evaporation, and place the plate on a shaker in a 37 degrees Celsius incubator for 24 hours at 50 revolutions per minute with a 15 mm orbit. At the end of the incubation, transfer the equilibrium bath from each well into individual polypropylene tubes, and make serial dilutions from the stock 30 micromolar cationic peptide carrier solution to generate a standard curve.